Hello everyone. Today we are going to discuss eukaryotic cells. In last video we discussed prokaryotic cells. The eukaryotes include all the protists, plants, animals and fungi. These cells have well organized nucleus and several membrane bound cell organelles. Genetic material or DNA is organized into chromosomes and chromatin. The various cell components of eukaryotic cells are described here to understand their structure and function. This is the picture of plant cell. Endoplasmic reticulum, nucleus, nucleolus, Golgi apparatus, nuclear envelope, plasma membrane, vacuole, middle lamella, cell wall, mitochondrion, ribosomes, chloroplast, cytoplasm, peroxisome, microtubules, plasmodesmata, smooth endoplasmic reticulum, lysosome. Cell membrane. Earlier studies showed that cell membrane is mainly composed of lipids, phospholipids, and proteins. In addition to phospholipids, membrane also contains cholesterol. Lipids are arranged parallelly within the membrane with the polar head towards the outer side and hydrophobic tail towards the inner part. Chemically, plasma membrane consists of lipids 20 to 79%, proteins 20 to 70%, carbohydrates 1 to 5%, and water 20%. Membrane proteins can be classified as integral or peripheral. Peripheral proteins lie on the surface of membrane, while integral proteins are partially or totally buried in the membrane. This is the important part. Many types of models are given to explain the structure of cell membrane. Danieli and Davison model. They proposed first lamellar model in 1935, in which they proposed that cell membrane contains four layers, two of phospholipids and two of proteins. Robertson model. Robertson 1959 modified the model of Danieli and Davison, a lipid bilayer being sandwiched between two layers of proteins on either side of it. An improved model of the structure of cell membrane are fluid mosaic model, proposed by Singer and Nicholson, 1972. This model is described as protein icebergs in a sea of lipids. According to fluid mosaic model, plasma membrane is composed of phospholipids, extrinsic or peripheral proteins, and intrinsic or integral proteins. The quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of membrane is important for functions like cell growth, formation of intercellular junction, secretion, endocytosis, cell division. One of the most important functions of the plasma membrane is the transport of molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is, from higher concentration to the lower. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is, from lower to the higher concentration. Such a transport is an energy-dependent process in which ATP is utilized and is called active transport. Example: sodium potassium pump. Cell wall. It is the non-living rigid, protective and semi-transparent outer covering present in plant cells, fungi and some protists. Algae have cell walls made of cellulose, galactans, mannans and minerals like calcium carbonate. In plants it consists of cellulose, hemicellulose, pectins and proteins. A cell wall has three parts: primary wall, middle lamella and secondary wall. Primary wall. The cell wall of a young plant cell is called primary wall. It is first formed wall of the cell. It is capable of growth which gradually diminishes as the cell matures. Secondary wall. It is produced in some mature cells when the latter have stopped growth. Example: trachees, vessels, elements, fibers. It is formed on inner towards membrane side of the cell. Middle lamella. It is layer mainly of calcium pectate, which holds the different neighboring cells together. The cell wall and middle lamellae may be traversed by plasmodesmata, which connect the cytoplasm of the neighboring cells. Functions: provides shape and rigidity to cell, protects protoplasm from mechanical injury, counteracts osmotic pressure and prevents bursting of plant cells by inhibiting excessive endosmosis. Important points: structural element of cell wall is microfibrils. Primary wall is the first formed wall of the cell. During ripening of a fruit, it becomes soft and pulpy due to dissolution of middle lamella. Plasmodesmata are the protoplasmic strands between adjacent plant cells and were described and named by Strasburger. Endomembrane system. It consists of membrane-bound organelles which function in close coordination with one another. Endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. Nuclear envelope is also considered a part of endomembrane system, as its lumen is in continuation with the lumen of endoplasmic reticulum. Endomembrane system is used to process short and recycle proteins with specific targeting of membrane traffic. Endoplasmic reticulum. Endoplasmic reticulum is a network of tubular structures scattered in the cytoplasm. They divide intracellular space in two compartments, that is, luminal inside ER and extraluminal cytoplasm. These can extend from nuclear envelope to plasma membrane. The endoplasmic reticulum is composed of the following three kinds of structures: cisternae, which occur in those cells which have synthetic rules; tubules, which are involved in lipid and steroid synthesis; vesicles, that are membrane-bound vacuolar structure. The ER is of two types: smooth endoplasmic reticulum (SER) consists mainly of tubules and vesicles. Rough endoplasmic reticulum (RER) consists of cisternae. It has ribosomes attached on its cytoplasmic surface.